Welcome to my update on BioInvent. I'm Martin Welschhoff and I'm the CEO of the company. On slide 2 you see our forward-looking statement, and I want to start with a quick company snapshot. BioInvent is a leading immuno-oncology antibody company focusing on immunotherapy by overcoming tumor resistance. We are highly integrated and cover the value chain from discovery, manufacturing until and including clinical development. With our unique core technology, we have generated a portfolio of programs with currently 4 programs in the clinic. This platform has been validated by a number of collaborations, most recently by Pfizer. We are listed on the Stockholm Stock Exchange and have a diversified shareholder base, including high-quality institutions such as Redmile, Van Herk Investments, Omega, HBM, Merieux, Swedbank Robur, Invus, Handelsbanken, and Consonance. 2020 has been a very successful year, and we have achieved several important milestones in clinical and preclinical development, and we also have been active in partnering. We have earned milestones in already existing partnerships, such as Daiichi and Pfizer, and established a new partnership with CASI Pharmaceuticals for 1206, exclusively for China, Macau, Hong Kong, and Taiwan. And last but not least, did a successful financing generating $70 million plus US in a combined directed and rights share issue. In 2021, we have been also very active so far. We presented exciting data for our lead program BI-1206 in a KOL event during the first quarter. We pushed ahead with our portfolio and started to enroll patients for BI-1808 and BT-001, and did another very successful financing of around $160 million US in a directed share issue. During the remainder of this year, we hope to initiate our 5th clinical program, BI-1607, and will remain active in business development and are planning for an update on BI-1206 in solid cancer in combination with pembrolizumab at the end of this year. At the core of BioInvent, we have our unique first platform for the discovery of targets and antibodies. We have established a close collaboration with a local hospital and receive human tumor tissue and cells on a regular basis. We screen our high-quality encoder human antibody collection against the fresh human material. Once we have obtained specific antibodies, we check their therapeutic effects in relevant models, after which we identify the targets. The output of this platform are preclinical validated targets and antibodies. Based on the first platform, we have established a portfolio of multiple value drivers. We have 2 antibodies targeting Fc-gamma2b. BI-1206 is currently in 2 clinical programs, one for non-Hodgkin lymphoma and one for solid cancers in combination with rituximab and pembrolizumab. As already mentioned, 1206 has been exclusively partnered with CASI for China, Macau, Hong Kong, and Taiwan. And we also have a supply agreement with Merck for Keytruda in the solid cancer study. We hope to initiate clinical development for the second anti-Fc-gamma2b antibody, BI-1607, towards the end of this year. We have an ongoing screening program on T-regulatory cells for the tumor microenvironment, which has generated 2 clinical programs so far. BT-001 is our proprietary anti-CTLA-4 antibody combined with transgene's oncolytic virus, and this is a 50-50 joint venture. BI-1808 and BI-1910 are antibodies targeting TNF receptor 2, which is one of the hot new targets in immuno-oncology. Both 001 as well as 1808 have started to enroll patients. Under our collaboration with Pfizer, we initiated a broad program identifying targets and antibody against tumor-associated myeloid cells, which are still at the preclinical level. BI-1206 is our lead program. By blocking Fc-gamma2b, 1206 enhances the anti-tumoral activity of other antibodies, such as anti-CD20 or anti-PD-1 antibodies. We are currently doing development for relapsed or refractory indolent non-Hodgkin lymphoma, patients who are resistant to rituximab, and advanced solid tumor patients who have relapsed or are refractory to anti-PD-1 and anti-PD-L1 therapy. In the non-Hodgkin lymphoma, we are currently focusing on 3 key segments: marginal zone lymphoma, follicular lymphoma, and mantle cell lymphoma. For mantle cell lymphoma, we also have the orphan drug designation. The phase 1/2 study is currently focusing on patients who have relapsed or are refractory and do not respond to anti-CD20-based therapy anymore. We are currently at the end of part A and will release data during Q1, which I will explain in more detail on the next slide. At that time, 15 patients were enrolled and 9 patients were evaluated. Out of the 9, we had 2 complete responses and 4 partial responses. Today, the complete responses are continuing 1 year and 2 years plus and are both at non-optimal doses. In the partial response, we also had a mantle cell lymphoma patient with blastoid histology, and we achieved a full depletion of the circulating tumoral B cells. The next milestones in this study are the selection of the dose for the part 2. We also want to include China in our global development plan, and we are planning to have an end of phase 1 meeting with the FDA during the 3rd quarter. We are also in the process of determining the quickest path to registration. In our second 1206 phase 1/2 study, we are focusing on patients with solid tumors who have relapsed or are refractory to anti-PD-1 and anti-PD-L1 therapy. We run this study under a clinical supply and collaboration agreement with Merck, and we are currently in part A and plan to provide an update towards the end of this year. The application potential for 1206 is broad and covers non-Hodgkin lymphoma, solid tumors, as well as autoimmune indications. We are focusing internally on oncology and are exploring the autoimmune space through collaborations. As already mentioned, we have a broad approach regarding 2 very interesting cell types of the tumor microenvironment: T regulatory cells and tumor-associated myeloid cells. Our goal is to identify targets and antibodies that either allow for the efficient depletion or reactivation of those inhibitory cells. From this approach, we have identified 2 very interesting programs: our proprietary anti-CTLA-4 and antibodies targeting TNF receptor 2, which is one of the hot and upcoming targets in cancer immunotherapy. BI-1808, our lead anti-TNF receptor 2 program, is currently in clinical development. We are exploring 808 as single agent and in combination. As a single agent, we are exploring non-small cell lung cancer, ovarian cancer, and CTCL. In combination with pembrolizumab, we will explore non-small cell lung cancer and ovarian cancer. It is interesting to note that we have been allowed to start the study at a relatively high dose. In collaboration with Transgene, we are developing our proprietary anti-CTLA-4 antibody, which is nicely differentiated from ipilimumab, since it's a more potent Treg depleting antibody. The concept builds on 3 clinically validated axes: anti-CTLA-4, anti-PD-1, and oncolytic viruses. And we expect enhanced efficacy and improved tolerability in a variety of tumor types, including metastatic melanoma, cutaneous melanoma, renal cell carcinoma, and non-small cell lung cancer. We have cloned the antibody into Transgene's oncolytic vaccinia virus, and the reagent is then applied intratumorally and leads to a very high concentration of the antibody in the solid tumor environment with a very low systemic exposure. We presented very strong preclinical data last year, and the study has started to enroll patients. I hope I could demonstrate that we have a strong portfolio with multiple value drivers, which represent multiple shots on goal, since each program represents first-in-class potential. Thank you very much for your attention.